Hi, so far we've discussed how by placing a subject in a scanner we can get a signal, and how we can use this signal to reconstruct an image. Last time we also discussed how in an fMRI experiment we collect a number of such images and study changes in oxygenation patterns across time. Finally, we talked about some of the characteristics of this MR signal and also about its noise. In this module, we're going to be discussing a little bit more about the structure of fMRI data. A standard fMRI experiment, as we've seen, gives rise to massive amounts of data. Now, in an fMRI experiment, we typically acquire both structural and functional data. The structural data is acquired for pre-processing purposes and also for presentation purposes, as we'll see as we move along. And so here we're going to discuss the structure of the data and some general terminology associated with it. We're also going to provide a brief overview of some of the characteristics of the functional data. When designing an fMRI experiment, we always need to balance the need for adequate spatial resolution with adequate temporal resolution. So temporal resolution, this is what's going to determine our ability to separate brain events in time. And in general, in fMRI, the temporal resolution is going to be determined by how quickly each individual image is acquired. And as we talked about in a previous module, this is determined by the TR. So this is how often we acquire a new image. The spatial resolution, on the other hand, that's going to determine our ability to distinguish changes in an image across different spatial locations. So as we saw at the end of the last module, when we move from a 32 by 32 image to a 64 by 64 to a 128 by 128, we can distinguish brain tissue and boundaries between brain tissues in a much better manner. And so the increased spatial resolution allows us an increased ability to distinguish changes in an image. In fMRI, we acquire both structural and functional images. And so the structural images are often these T1 weighted images. And so they're high spatial resolution, so you can make out fine anatomical detail by looking at them. However, they have no temporal resolution because they're only static images. But using these images, we can distinguish between different types of tissue and make determinations about which brain regions are located where. Functional images, on the other hand, are T2 star weighted images. And they have significantly lower spatial resolution, but a much higher temporal resolution. We actually acquire these images every 2 seconds or so. And using these types of images, we can relate changes in signal to some experimental manipulation. So for example, if we're performing some condition, which we'll call it condition A, we can look at images that are collected while we're performing condition A and compare them to when we're performing some other type of condition, say condition B. And we can assume that, say, condition A here is finger tapping and condition B is rest. So is the signal higher in a certain voxel when we're finger tapping versus when we're resting? Here's some more terminology. So when we look at the brain, it's hard to visualize it in 3 dimensions sometimes. So sometimes we look at single slices of it. And so depending on which direction you slice the brain, we have either coronal, sagittal, or axial slices. And so MRI images are typically acquired in axial slices, one at a time. So this can either be performed in a sequential manner, where we perform one slice after the other in order, or in an interleaved manner where we first collect the first slice and then the third slice and the 5th slice, etc., and then go back and do the 2nd, 4th, whatnot. That's called interleaved sampling. But once we have all the slices collected, we combine them, and this makes up a 3-dimensional brain volume. So the field of view of an image is basically, you can consider that you have like a box around a person's brain, and you're imaging within that box. The width of that box is called the field of view, or the FOW. Now, within the field of view, we sample a bunch of slices, and each slice has a thickness as well. And so here we illustrate that we have a slice thickness of 3 millimeters. So each slice is 3 millimeters, then we move to the next one, which is 3 millimeters, etc., etc., etc. We also have a resolution in the slice, and this is typically referred to as the matrix size. And so in this example, we say that we have a 64 by 64 image, and this means that we have 64 voxels in both the X and the Y direction. Now, if you divide the field of view by the matrix size, you get the size of the voxel. So in this case, if we have a 192 millimeter field of view and 64 voxels in that direction, then each voxel is going to be 3 millimeter. So in this little cartoon here, we have slice thickness 3 millimeter and in-plane resolution of 3 millimeter. So our voxel size is ultimately going to be 3 by 3 by 3. So in general, in an fMRI experiment, we study many subjects. And each of these subjects may be scanned during multiple sessions. And in each of these sessions, they can consist of multiple runs. So we might repeat the task or the experiment several times while the person is in the scanner. And each of these runs consists of a series of brain volumes, because each run we might collect several hundred brain volumes. As we saw here, each volume is made up of multiple slices. Each slice contains many voxels, and each voxel, that's our fundamental unit of measure here, has an intensity associated with it. So this kind of illustrates the hierarchy of the data we have. So we have the experiment, and in the experiment we have subjects. Within the subjects we have sessions, runs, and within each run we have multiple brain volumes. So again, if we look at these multiple brain volumes, we can extract data from a single voxel and look at that, and then we have a time series. So basically what we have here is just on this level we have a multivariate time series problem. But really what we have is a multivariate time series problem that's very hierarchical. So this is just the data for a single subject, single session, single run, but we might have a hierarchy of such data. So now I want to give you kind of a roadmap of where we're going from here. So far in the previous modules, we've really talked about data acquisition and reconstruction. In the next couple of modules that are coming up, we're going to move towards experimental design and pre-processing. So we're going to cover that in the next week's modules. After that, we're going to move on to data analysis, and we're going to spend a good amount of time on each of these topics of data analysis. And we'll first talk about localizing brain activation, then we'll talk about connectivity, and then we'll talk about prediction. Okay, that's the end of this module. Here we've discussed a little bit about the structure of fMRI data. In the coming modules, we'll talk a little bit about experimental design and on pre-processing before we move on to the statistical analysis of fMRI. Okay, see you in the next module.